with the ICI, and 17 alpha was not blocked by ICI.
Well, for these receptors, it's not blocked, right? And then, so we don't think that it's ERX, and we actually eliminated this because one ICI 182, 780 blocks in our hypothalamic astrocytes, and 17 alpha doesn't activate. So we automatically ruled the last one out. So the other four we're actually going to study further. And so the first thing we did is a dose-response curve with several different agonists. PPT is a selective estrogen receptor alpha agonist, and it has preferential binding to estrogen receptor alpha over beta about 410-fold. So these are not pure agonists. And then DPN is a selective estrogen receptor beta agonist, and it has preferential binding of estrogen receptor beta about 70 times higher than estrogen receptor alpha. Then you have STX, which is the selective estrogen receptor modulator that activates the STX binding protein. And then you have G1, which is a GPR30 agonist. So we wanted to see how the different agonists respond in the hypothalamic astrocytes to see which one is the closest to what estradiol does. So in the yellow is the dose-response curve that I showed you earlier of estradiol with an effective concentration, a half maximal effective concentration of 0.15 nanomolars. And so with PPT, which is a estrogen receptor alpha agonist, we see that the response is here. The maximum response is very similar, but at lower concentrations, it was a little bit more potent. And so the EC50 was a little bit lower at 0.03 nanomolars. Then we looked at DPN, which is estrogen receptor beta agonist, and we see that it's actually a much weaker agonist, and it requires about 100 times higher concentration in order to get the response going. But the maximum response is very similar, but it's much weaker. And then STX is in the bluish gray here. So very similar response, but at the higher concentrations is actually a little bit more effective. And then there's G1, which is a GPR30 agonist. And again, that's very similar to the DPN, where it required much higher concentrations for their similar effect. So I just wanted to point out that DPN and G1 are not very similar to, the response is somewhat different than what estradiol looks like. So physiologically, it looks a little different, and it required much higher concentrations. So therefore, for the progesterone assays, for DPN and G1, we actually used 100 nanomolars, as opposed to 1 nanomolar for estradiol, PPT, and STX. So I just wanted to show the rationale for using different concentrations. In our progesterone study, we wanted to see that these agonists will produce progesterone. And so with estradiol, we got a good progesterone response, and we've seen that also with PPT and STX and also G1. But DPN, even at 100 nanomolars, did not stimulate progesterone production. So this tells us that it's probably not signaling through estrogen receptor beta. And then we know it signals through a glutamate receptor, and so therefore we block the mGluR1A with LY36738-5, and then we wanted to see if this would block the different signaling pathways. So estradiol again was blocked. PPT was also blocked, which is estrogen receptor alpha. The STX was also blocked. DPN did not respond to it did not respond with the progesterone synthesis. But what's interesting is that the G1 could stimulate progesterone production, but actually was not blocked by LY36735. So this tells us that there's probably a different pathway or some different signaling mechanism for GPR30 as opposed to estrogen receptor alpha or estradiol. So for our calcium studies, we wanted to repeat the same thing and see if LY36738-5 would block. And actually for all four, PPT, DPN, STX, and G1, here G1 actually, it actually blocked the calcium response, but not the progesterone synthesis. And then we wanted to see what would happen if we added the DHPG in addition, this is DHPG at 100 nanomolars in addition to the different agonists. And we see that there was an increased response in PPT, DPN, STX, but G1 actually did not increase. So again, this is a little bit different than the other agonists. So what does this mean? So summary at this point, PPT and STX are the most closely mimicking estradiol's effects, which suggests that the estradiol may signal either through membrane-associated estrogen receptor alpha or an STX binding protein. DPN failed to stimulate progesterone synthesis and required high doses for calcium release. Thus, estradiol is not signaling through membrane estrogen receptor beta. And then GPR30 probably signals through its own unique pathway, which we'll talk about a little further. So the next step was to do some co-immunoprecipitation And in these studies, what we want to do is to see if two proteins interact with each other. So we would lyse cells, and then we could either study the whole cell or just the plasma membrane fraction. And then we would add an antibody to that lysate or fraction, run it on a column that recognizes the antibody so that you can isolate the protein of interest. And then you can elute the column and then run it on a Western blot, and then on the Western blot you probe it with a second antibody. So if the first protein and the second protein are attached, it'll stay on in the column, and now it'll be on the Western. And so when we probe with estrogen receptor alpha, so the first one was mGluR1A, and then after we run on the Western, we try to see if it's attached to estrogen receptor alpha. And we see that there is some association in the whole cell, but it's mostly the association on the plasma membrane fraction, which makes sense because the mGluR1A is a membrane receptor. Then when we looked at estrogen receptor beta, we actually did not see any interaction in the whole cell or on the plasma membrane, which again probably specifies that the glutamate receptor and the estrogen receptor beta are probably not interacting. This is probably not the main mechanism. What was interesting is that when we did this co-immunoprecipitation for mGluR and GPR30 down here, we actually did not see any interaction, which again signals that it probably signals through a different pathway. But then when we looked at GPR30 and just a regular Western blot, we could find it in the whole cell, in intracellular, but we can't find it in the plasma membrane. So this is kind of consistent with other literature where nobody has actually been able to show native GPR30 on the plasma membrane. They've only been able to show it on the smooth endoplasmic reticulum and the Golgi apparatus. So again, GPR30 may be a little bit different signaling mechanism that actually may involve intracellular signaling. So summary of findings: estrogen receptor alpha, but not estrogen receptor beta or GPR30 interacts with the metabolite glutamate receptor 1A, as demonstrated by co-immunoprecipitation. GPR30 was not identified on the plasma membrane. And so our hypothesis for G1 for the GPR30 signaling is that since G1 stimulated progesterone synthesis was not attenuated by LY36735, it suggests that the G1 may directly activate an intracellular GPR30, such as on the smooth endoplasmic reticulum, to directly release the calcium, which can then stimulate progesterone production. Now, G1 required high doses for calcium release, which may make more sense because it has to go into the cell to signal. But then again, from our calcium studies, the LY36735 blocked G1's effect, but it was not enhanced by DHPG, so that also just suggests that instead of acting on a GPR30, which goes through mGluR1A, it actually may also activate mGluR1A. And so the G1 and the DHPG would act on the same membrane and compete against each other. You wouldn't get a higher effect. But in that, calcium can actually be blocked.
What's the chemical structure of G1? Can it cross the membrane? I mean, is it lipid-filled?
Exposed.
Yeah. Yeah, I can't remember the actual structure. Sorry. So the next step was to basically try to study the estrogen receptor alpha and the STX, which are the two receptors that we kind of narrowed down. So the next thing we did is to take estrogen receptor alpha knockout mice, and the first thing we wanted to do is to this is actually an in vivo study, and these are oophorectomized female rodents, either wild type, and when wild type is stimulated with estradiol, and then we look at the hypothalamus, there's an increase in progesterone production in the hypothalamus, which goes along with the LH surge. But in estrogen receptor alpha knockout mice, there was no increase in progesterone. In the literature, the estrogen receptor alpha knockout mice actually don't have positive estrogen feedback, they don't have LH surges, and they don't have sexual receptivity. The lordosis quotient is almost 0. So estrogen receptor alpha is very critical for estrogen positive feedback and for sexual reproduction.
John, and I'm not aware, do these ovulate at all?
No.
They don't have spontaneous.
No, they don't have estrogen positive. They never have an LH surge. So
But if you give them exogenously to ovulate, with gonadotrophin, do they ovulate? I'm just trying to think whether it's a central mechanism or a peripheral, just for my curiosity.
I don't know if there's any studies on giving gonadotropins. I think it's more of a central mechanism, but I don't know if it's also in the ovary. I just know that there's no estrogen positive feedback, so it's never a trigger. But that's interesting. So this is consistent that the wild types can have a progesterone response after estradiol injection, but not the so in vitro, we took astrocyte cultures of the wild types and compared them to the ERCOs. And we see an estradiol will stimulate a calcium response, but in the ERCOs, this calcium response is attenuated, and the calcium response was not significantly increased. So this tells us that estradiol mainly signals through probably the estrogen receptor alpha. Also interesting was the DPN at 1 nanomolars, again signaled a little bit in the wild types and a lot more at 100 nanomolars, but this was actually abolished by estrogen receptor alpha knockouts. So therefore we think that, again, the DPN is not a pure estrogen receptor beta agonist. It actually, it's about 70-fold higher preference for estrogen receptor beta. So at 100 nanomolars, it can actually also activate estrogen receptor alpha at about a 1 nanomolar response. So that's probably what's happening here, is that estrogen receptor alpha is being activated by DPN and not beta. STX and G1 are actually fully able to signal through estrogen receptor alpha knockout mice. So this demonstrates a few things. One, it demonstrates that there's probably a different signaling mechanism. It's independent of estrogen receptor alpha. But also, two, that even though this signals and it has a very high calcium level, it may not be relevant in reproduction because these mice actually don't produce progesterone and they don't ovulate. So summary from the ERCO mice is that the estradiol-induced calcium release was abolished, which supports estradiol signaling through membrane estrogen receptor alpha, and it's consistent with lack of estrogen-positive feedback and lack of sexual receptivity in ERCO mice. DPN at 100 nanomolars did not stimulate the calcium release, which suggests that DPN acts upon membrane-associated estrogen receptor alpha and not membrane-associated estrogen receptor beta. STX induced a similar calcium release compared to the wild types, which suggests that the signaling pathway is independent of MER alpha, but it's of unclear physiological relevance and function. And then G1 induced calcium release, which was not blocked, so that's still consistent with our hypothesis that it could signal on the plasma membrane for mGluR1A, as well as activate directly intracellular GPR30. So overall, we believe that membrane estrogen receptor alpha is the main receptor responsible for rapid estrogen signaling in hypothalamic astrocytes that's involved in estrogen-positive feedback and the LA surge. So the last part of the talk is going to focus on sex differences in hypothalamic astrocytes. And if we compare male and female astrocyte cultures, the females that we've been studying so far respond with a calcium increase. The males actually also increase calcium, but it's at a lower level. So that's interesting that the males can respond. However, that when we look at the progesterone, we use 1 nanomolar, and the female will release and synthesize and release progesterone, but the male mice and male rats actually don't increase progesterone production. And then we tried it with 100 nanomolars, and again, the estrogen can induce progesterone synthesis in females, but not in the males. So again, this may also tell us that there may be some threshold that this lower calcium may not be enough to stimulate progesterone production. But it's unclear if it's, if these mice had a high enough calcium, whether or not they would produce progesterone. So that would be the study. So I just wanted to talk briefly about surface biotinidation because one of my studies involves this process. So there's commercially available kit, Sulfano NHS biotin, and the biotin is a large molecule. It's water soluble, but it's membrane impermeable. And with this, you can actually cause reactions to primary amines, either on amino acids or on an N-terminus, if they stick out of the plasma membrane. So anything that sticks out of the plasma membrane, this can form amide bonds if it has a primary amines. And the purpose is to demonstrate if membrane estrogen receptor alpha has an extracellular portion that sticks out of the membrane, and also it can tell us if estradiol levels can actually regulate the insertion and internalization of membrane estrogen receptor alpha. So I'm actually not going to talk about the internalization studies, because that kind of goes off my talk, but I'm going to just talk about insertion really fast. So if you have an attack cell, you have some membrane, in this case estrogen receptor alpha, and then you incubate with estradiol. We want to see if this would actually cause an increase or decrease of membrane estrogen receptor alpha levels on the membrane. Then, after incubating, we would biotinylate, and then which forms stable amide bonds, and then we want to wash off the excess free biotin. And then we take these cells, we lyse them, run them on a column that recognizes the biotin, so anything that is sticking to the biotin is on the column, and then we elute the column, run it on the western. So this is not specific binding, it could be any protein or membrane receptor on the plasma membrane, but then we probe this for our membrane receptor of interest, which is estrogen receptor alpha. So I participated in the study. This is not my primary study, but we can actually see that at about 30 minutes of estradiol exposure, that there's actually increase in estrogen receptor alpha concentrations on the plasma membrane, as well as an immunoreactive, which we believe is a splice variant of estrogen receptor alpha. Actually, and they both increase in parallel and then decrease, and at 24 to 48 hours, it's actually back at baseline levels. So this tells us that the estrogen is regulating the membrane estrogen receptor levels on the plasma membrane. And I wanted to talk about that because we actually did an experiment. We know at 30 minutes, there's the maximum amount of estrogen receptor on the plasma membrane. So we looked at males and females at control, and we see that males and females both have estrogen receptor alpha on the membrane, and the females were not significantly higher than the males. And then after about 30 minutes of estrogen exposure, in the females, the amount of membrane estrogen receptor increased, which was statistically significant. The males increase was not statistically significant. So this tells us that maybe this is some mechanism where there may be more response of estrogen receptor alpha going to the membrane, and maybe being able to generate a higher calcium response.
Did you see with the other agents which release this calcium, whether there's a difference in the male and female?
We actually didn't study all the other compounds. We didn't study the other agonists in the males.
No, because one you talk of receptors, but it may be a fundamental difference that they just don't release more calcium, right?
Right.
So
It's
that
possible.
would be, yeah,
Yeah, it may not be a threshold, it may be some different.
yeah.
I mean, John, just to add on that, I know at least in human GVO-site, androstenedione antagonizes the ability of estradiol to mount calcium flux, and I'm wondering, are some of these sexual dimorphic changes due to androgens?
You mean like earlier on in development and it changes the cell?
Well,
Or-
perhaps, but even acutely. In other words, if you co-incubate a GVO site with estradiol and you induce these calcium oscillations, you can block them with androstenedione. So you could actually posit an acute effect as well.
Well, these are all in culture, so I mean, there's no effect because we actually incubated with charcoal strip media. I mean, a charcoal strip serum before, so they weren't exposed to estrogen. And then we actually added only estrogen in it.
But can you antagonize some of your data in the female work with androgens?
That we never tried. Yeah. So the last part of the study is we wanted to see if these differences in the hypothalamic response, in the hypothalamic estradiol response is due to either hormone or chromosomal effects. So it could be chromosomal effects from the X and Y chromosome, and basically, the females have X and X and the males have X and Y, and we wanted to see if that was a main effect, or if it's more of a gonadal effect. So the SRY is the sex-determining region of the Y chromosome. So if you have the SRY on the Y chromosome, it will basically shift the development of the gonad into testes, and if you have no SRY, the gonads will develop into ovaries. And so if it's a gonadal effect, you can have two types of hormone effects, either organizational effects, where this difference in hormones and the sex hormones earlier on in development can actually change the cells, cause differentiation of the cells early on in development. And then, for instance, in the rodents, once estrogen positive feedback is gone, it never comes back. So if you take males and then you try to get an LH surge in rodents, in the humans you can, but in rodents, they actually will never have a positive estrogen feedback. And then you can have activational effects, where you're basically, at the time of your experiment, you can add certain hormones and have certain effects from those hormones at the time of your experiment. So we wanted to study if some of these sex differences were due to hormonal effects versus sex chromosome effects. So for this, we used 4-core genotype mice from Art Arnold. These mice are actually obtained from using regular females, and then the males actually have an SRY that's taken from the Y chromosome and basically transgenically shifted onto an autosome. So they have an SRY and they have a Y chromosome, but the SRY is not on the Y chromosome. And so these are males, and then when you cross these two, you can get 4 offspring. You can have regular females, XX females that have ovaries. You can have XY females, so the X and the Y, but the Y doesn't have the SRY, and it results in ovaries. So you have XY females. And then you can have an XX with an SRY and autosome, which will develop into testes. So then you have XY males. And then you can have these, basically the same thing, you can have XY males. However, these are not the same as wild type males. And then you can compare these groups. So if you have ovaries, regardless of XX or XY, or testes, and there's a difference between these two. It's a sex effect. And then if you have basically XX or XY, regardless of ovaries or testes, you can have a chromosome effect. So we can compare the 4 groups. So what we did is we did progesterone assays, and then we incubated these 4 different astrocytes types cultures with estradiol at 1 nanomolar. And we see that in the animals with ovaries, the XX females and the XY females, that there's a significant increase in progesterone production stimulated by estrogen. And then in rodents that had testes, regardless if it was XX or XY, there was very little increase in estrogen production. So it's mostly an effect between these 2 and these 2, so it's a pretty large sex effect. There is some difference in the baseline of the progesterone, so it hints at maybe some possible chromosomal effect. But if you remember a few slides ago, even wild-type males actually produce some progesterone, and so these male rodents actually are somewhat probably different than the phenotypically wild-type males that actually do have some progesterone response. These actually haven't been characterized, but there's actually been reported of a lot of differences in terms of function and things in these rodents versus wild-type males. So the summary of the findings: estrogen-positive feedback occurs in female rodents, but not in males, and astrocytic sex differences in estradiol-stimulated calcium elevation and progesterone synthesis could help explain some of these differences. Membrane estrogen receptor alpha is an integral membrane receptor with an extracellular portion that's not associated with the intercellular leaflet. So earlier on, it was believed that maybe this receptor is actually cytoplasmic and associated with the membrane only and doesn't stick out, but it seems like we are able to bioattenuate it. And then sex differences in membrane estrogen receptor alpha trafficking could contribute to greater estradiol responses in female astrocytes. And these astrocytic sex differences are most probably due to differential secretion of gonadotropins. However, the SRY itself can actually also have a function, and so we can't completely rule that out. So in conclusion, estrogen receptor alpha is an integral membrane